Before we proceed, we will also like to take a moment to thank the School of Science. This event will not have been possible without the work and support from Dean Mavalvola, her staff in her office, and then other members of the School of Science, including the professors here today. So thank you very much for your help with this. So I was looking at Professor Joy Davis's research website, and he has this quote in there by Charles Lindbergh that says, "The construction of an airplane is simple compared with the evolutionary achievement of a bird. If I had to choose, I would rather have birds than airplanes." So I'm going to guess he likes studying complex systems. So it wasn't a surprise to learn that his research focuses on, and this is a mouthful, massive macromolecular complexes that are essential in virtually all cellular processes, and uncovering how these complexes are assembled, disassembled, degraded, and how environmental stress, mutations, disease, and aging alter everything. So it's like very simple stuff, right? He earned his bachelor's degrees in computer science and biological engineering from UC Berkeley, a PhD from MIT in 2010, and then he joined the faculty in 2017. Please join me in welcoming Professor Joy Davis.
Awesome, thank you. Yeah, it's so nice to be here and talk about some of the work. So I wanted to start by framing this talk around a quote from my favorite physicist, probably many of yours, Richard Feynman. This is from a talk he gave in the late 50s called Plenty of Room at the Bottom. And he's sort of describing what are the key questions in biology at the time. These are things like what happens when DNA is damaged with obvious implications in cancer. Questions like how are proteins synthesized, that is, how does a ribosome sort of read a message and decode that into a protein that then can go and execute some function. That function might be something like photosynthesis, how do cells sort of convert light into chemical energy. These are many of the problems that we still wrestle with today. And he goes on to say, it's very easy to answer many of these fundamental questions, you just look at the thing. And I love this sort of idea that if we could look at atomic resolution at the natural world, we could begin to understand how these processes occur. Feynman then laments, unfortunately, the present microscope skis at a scale that is just a bit too crude. If we could make these more powerful, many of these problems would become easy. So what did these sort of structures look like at the time? This is a micrograph from George Pilates' lab in 1954. This is a eukaryotic cell, so there's a nucleus here. And each one of these little black dots is a ribosome. Okay, it was called the particles of Pilate at the time. They didn't even know that they were ribosomes because the resolution is so poor. If you jump forward 15 years or so, this is now a micrograph from Miller Lab, and now you can actually start to see a little bit more structure. So each one of, I don't know if this is working at all. Each one of the sort of lateral things emerging, those are ribosomal RNAs that are eventually going to fold up and form a structure. And if we go forward a little bit further, now we can start to see individual particles on the bottom row there. Those are individual ribosomes. And from a bunch of those pictures, people could infer a 3D structure. And those are sort of shown as these clay-looking models on the top. What I love about them is they sort of look like these artifacts of antiquity, right? They're really low resolution. You get a general sense of the shape, and maybe you could sort of guess what they might do, but you would never be able to see the molecular detail that you would need to really understand how these machines work. Okay, and so basically from the late 70s until very recently, this field called electron microscopy was sort of disparaged by structural biologists. It was called blobology. There was sort of a niche field. A few people were working in it, but nobody ever thought you'd actually be able to understand molecular mechanism using electron microscopy. And there was really a revolution in the last 15 years that have gone from this sort of blobology shown on the left to these atomic structures that we now use to regularly answer questions about the sort of processes that Feynman presaged in the beginning of this talk, right? So I just pulled a few random papers over the last few years, things about understanding how DNA damage gets repaired, how the ribosome translates messages into proteins, how photosynthesis occurs. And all these structures are solved by this process called electron microscopy, and specifically cryo-electron microscopy. So how does that work? So we're going to look at a quick movie here. This is starting with an electron microscopy grid. It's sort of a metal grid. This one's made out of copper. You can see there's these grid bars running across it. It's about 3 millimeters across. You can hold it with tweezers. I could walk around the room with it. And what I'm going to do is I'm going to zoom in about 30,000 folds, first starting in sort of the light regime and then switching over to the electron microscopy regime. So we're going to zoom in, and you'll see sort of between these grid bars now in the EM range, there is a thin layer of carbon, and into that carbon we've etched small holes. These are about a micron apart. And they're so small that you can suspend a thin layer of ice across those holes. And within that ice are individual protein molecules. So what you're looking at now are individual protein complexes imaged with an electron beam, and we can see isolated particles. So what do we do with this kind of data? We're going to lean on a lot of the machine learning tools that were developed for computer vision. If you think about self-driving cars, they need to recognize the environment around them, isolate what is a car, what is a person, what is a stoplight. We can repurpose some of those types of tools to now pick out individual particles. We'll extract those, and a typical data set we could collect here on campus overnight would result in tens of thousands to tens of millions of isolated particles. Okay, so you can see that the individual images of these things are still quite poor resolution. And that has to do with the fact that this is a biological sample. We're exposing it to ionizing radiation that causes damage, and so we have to expose it to a very low dose. And as a result, the individual images are quite poor. But we have lots of them. And the great thing about having lots of things is that you can average across them if you make the assumption that they're all the same. So that's exactly what we'll do. We'll sort of figure out what angle we've taken each one of these pictures from. We're going to sum up all of the images from that same picture. That'll give us a better one. And from that, we can infer a 3D structure. This all seems great. Those first 3 papers that I showed you, that's largely what they've done. I have a young child at home, and I sort of like to think about what does biology really look like. There's this one very cute static picture that you think about biology. And so by analogy, this is my son Will. You might think Will is a protein molecule, and that spoon is some ligand, and we're going to try to do a molecular docking experiment, right? So we're going to try to find the ligand binding site, and if you look at this static picture, it looks like a straightforward sort of task to complete. But in reality, biology is extremely dynamic, right? There's all these motions that are going on, and that's just an inherent part of the biology. The problem with this is when we've frozen these protein complexes in ice, we've sort of trapped Will in all these different states. Right, so if I just took this movie and thought of each frame as a different picture of Will on that EM grid and averaged across it, what I get back is this blurry sort of blobology-like vision, right? So you sort of have 2 approaches. One, you can biochemically isolate one state and sort of force things to be in one conformation. The challenge with that is that you've thrown away all the beautiful dynamics that support life. Okay, so really the mainstay of my work has been led by 2 fantastic graduate students, Ellen Zhang, who was my first student and now has her own group at Princeton, and this work is continued by Maria Carrera, another grad student in the MIT biology program. The idea was to take more tools from machine learning and figure out how we could better leverage these data sets to extract these molecular motions. We did that by putting together a set of networks we call an autoencoder that consists of 2 neural networks. The first one here is a decoder network in green. Its job is to try to reconstruct a particle image that we give it, okay? So we're basically gonna give it an image and we say, make a copy of that. If it makes something that looks the same, we say, good job. We leave the network weights alone. If they're different, we can use that difference to propagate a loss back to the network, and based on that loss, we can update the weights. How do we deal with the heterogeneity? We'll couple that to an encoder network. The job of this is to sort of sort the particles based on their structure into groups that are the same versus groups that are different. Okay, and if it does a good job sorting that, then the decoder can reconstruct them accurately. I've obviously glossed over some details. What I want you to take away from this is if we have a data set, we can train these models, and with a trained model, we can then project the particles into a low-dimensional space, and they sort of sort based on what's similar and what's different. So in this particular data set, you might say there's on the order of like a half dozen major structures. Those are sort of the yellowish colors, the high-density regions. But those interconvert between them because they're molecules in motion. Okay. We can use these to do a bunch of different things. We can extract individual states of the complex. We call these discrete states. We can walk along the data manifold and see the molecules in motion. We can extract particles and do other sort of tasks with them. Okay, so we built this tool. We began thinking about how we might apply it. And the first place we looked was at the spliceosome. This is a large, well-characterized structure. It was actually sort of worked on here at MIT in Phil Sharp's group. If you image it, you sort of get a molecule which looks highly resolved in some regions, sort of shown in blue. And then there's this other region in red where it's very hard to see what's going on. It's blurry. And so we hypothesized that is likely in motion. And if that was in motion, then a tool like CryoDragon should help resolve it. And that's exactly what we see. So if we train this model, instead of now seeing this blurry static thing, we can actually see the molecule sort of moving around as we walk along this data manifold. So this worked quite well with isolated particles. The next question was, if we can sort things based on their structure, perhaps we could image more and more complex mixtures of molecules and allow a tool like CryoDragon to sort them out. And so a postdoc and 2 other wonderful students in my group really took on this task. And their idea was to just sort of take this to the extreme. What if we lysed cells and took this complex mixture as a lysate, put that onto a grid and imaged it, and then used CryoDragon to sort of sort out all the structures? Well, how would we test that? We began to think about translation. This is a sort of system that we worked on a bunch. And what's amazing about it is over the last 20 years or so, many, many groups have worked on trying to determine what are the key structural states that support the translation by the ribosome. And that's sort of summarized here. This is probably 100 human years worth of work. If all these states are important, then they should all exist in the cell. And if they all exist in the cell and our tool works as well as we hope it does, we should be able to see them if we just put lysates directly on the grid. And of course, I wouldn't be telling you this if this didn't actually work out. So I'm just going to show you sort of an initiation cascade, where here you have a small ribosomal subunit with a couple factors bound. We can find the next state where another factor is bound, and the final state where this is now ready to initiate translation. We can go and look through the elongation cycle, so we can see sort of tRNAs moving through the peptidyl-transferase center, allowing translation to occur. And coupled with that, there's actually motions of the 2 subunits relative to each other. So this is how translation's occurring in the cell. We went a little bit further and we began to think, well, maybe we can do discovery science with this. So what if we have some cellular system, we don't understand something about how it works? In this case, we were interested in understanding how an archaeal organism, this single cellular organism, senses starvation and turns off ribosomal translation. All we had to do was grow cells until they were starved, lyse them and put them on grids. When we determined structures, what came back was some unknown density that wasn't part of the ribosome. So the interesting thing about this is that you now have a structure, but you don't know what protein is in there. It's sort of the opposite problem that you typically have in biology, where you start with genetics and then you go to biochemistry. Here, we have a structure, but what is it? What was amazing is that we could resolve it at high enough resolution that you could actually see side chains. So these are the side chains that are encoded in each one of the proteins. And we used a tool, another machine learning tool called Model Angelo, that basically built a sequence into this density de novo. It didn't know what the sequence was, it didn't know what organism it was, but it just builds it in directly. But now we have a sequence, and we can use that sequence to now go search in the proteome of that organism, and we get a specific hit for it. And so that hit ends up being this translation silencing factor that we're calling AHA. This was our AHA moment that this worked. Okay? And what was cool about it is that not only do we now have the gene that's important for this, but we can infer mechanism of action because we see it bound to its target. Okay. The last thing I want to tell you is that sort of the future of where this is going, which is now starting to look at structures directly in cells. What I've shown you so far was first with purified material, then what we're calling sort of ex vivo with lysates on grids. What we'd love to do is look directly how these work in cells. And so Barrett Powell is a final graduate student in my lab I'll describe today who worked on this. He developed a tool we call TomoDragon that allows us to directly look at structures in cells. And so I'm going to show you is sort of a slice as we move through a bacterial cell. This happens to be E. coli. What you'll see as this plays is that on the sort of left side of the screen, or your right side of the screen, that's the plasma membrane. And then all those white splotches, those are ribosomes. Barrett's now going to populate those. Those are all in different states. And you can see there's always this joke that E. coli is a bag of ribosomes, and it often just looks like a bag of ribosomes when you look at these. What's so neat about this is we can sort these based on structure that allows us to average these things, and now we can get high-resolution structures of ribosomes directly in cells. The sorts of things we can do with that is that we can see these individual structural motions. And then there was a little trick here. These cells have been treated with a small molecule drug called chloramphenicol. It binds to and inhibits translation. And what we were so excited to see is we could find particles where we could actually build in density for this small molecule. So this is a therapeutic bound to its physiological target and directly imaged in the cell. With that, I'll just sort of conclude with what I've shown you. I think that these new tools in cryo-EM are allowing us to realize the promise of structural biology, to really understand the natural world at atomic resolution by looking at it. We can do that both in vitro, with purified samples, with lysates, and then going forward, we're looking to do this in the cell. Thank you.